Let's talk about Lab 7, Photosynthesis in Spinach Chloroplasts. Lab 7 has two parts. We're going to make an absorbance spectrum and an action spectrum. So let's talk about what those things mean. In this course, we have observed chloroplasts, the site of photosynthesis, in Elodea cells and also in the protist euglena. So there's a photograph right there of some euglena cells, and there are the chloroplasts. So here's a little schematic from the textbook of the chloroplasts, and you can see the green part, that's where the pigments are found. In today's lab, photosynthesis will be examined in more detail in isolated spinach chloroplasts. In the first exercise, we will be investigating the absorbance of the pigments. There are many pigments found in chloroplasts. You may be familiar with some of them, such as the chlorophylls. There's chlorophyll A, which is a blue-green color, chlorophyll B, which is a yellow-green color, and there are lots of other pigments, such as the carotenes, which are orange and red colors. We have the xanthophylls, which are yellow, and there might even be some purple xanthophylls, if I remember correctly. We also have this one here, phaeophytin A, which is kind of a grayish-brownish color. And we have phycocyanin, which is a bluish color. So many pigments out there that are found in the chloroplasts. Each pigment has a unique absorption spectrum. So you're probably wondering what an absorbance spectrum or an absorption spectrum is. This is where basically we take the pigment or chloroplast or whatever it is and we put it in a spectrophotometer and we see which wavelengths it absorbs best. So we can do this quantitatively. So if you take a look at this particular graph, we have a whole bunch of pigments from chloroplasts have been put through a spectrophotometer and their absorption spectra are put on the graph. If you take a look at something like chlorophyll A and chlorophyll B and C, they absorb really well in the blue and a little bit less so in the red region. This is why they look green, because they absorb all the other colors. Whereas the other pigments maybe do different things. The carotenes absorb really well in the blue and green areas, but not so much in the yellow and red areas, so they appear orange. In part A of today's lab, we will generate an absorption spectrum of the spinach chloroplast to determine which wavelengths of light the pigments in the chloroplast absorb best. So this is very easily done. Basically, all you need is some spinach chloroplasts, which I have there on the left, and we need some buffer. This is what the chloroplasts are in. We're going to use that as a blank, and we need a spectrophotometer. So if you take a look at the table on the right, what we're going to do is basically measure the absorbance for the spinach chloroplast suspension at all of those wavelengths there. So starting at 400 nanometers, and we're going to increase by 20 nanometers and again, and we're going to cover the blue range of the spectrum. Then we're going to cover the green range of the spectrum, so from 480 to 540 nanometers. And eventually we'll make it all the way up to the red part of the spectrum, which goes from about 660 to 680 nanometers. So at this point, that is part A. You're going to take that information and you're going to make a plot. So it'll be an absorption spectrum. On the y-axis, you'll have absorbance. On the x-axis, you'll have the wavelength. It is assumed that the wavelengths of light that are maximally absorbed are also the wavelengths of light where we are going to have the highest rates of photosynthetic activity. And this leads into part B. In the second exercise, we are going to expose these isolated chloroplasts to 3 different wavelengths of light, so blue and green and red, and we're going to indirectly determine photosynthetic activity. This type of experiment will produce an action spectrum, which will indicate those wavelengths of light that will produce the highest rates of photosynthesis. To understand an action spectrum, it is best to know a little bit about the reactions of photosynthesis. So photosynthesis occurs in 2 stages, the light reactions and the Calvin cycle. So let's just review those now, and we of course are going to go over them in much detail in the lectures. So the light reactions of course require light and they require water. They also require ADP, inorganic phosphate, and NADP+. So basically what happens is light is going to strike the thylakoids. This is where the pigments are found, and then it is going to split water. So this is going to release oxygen, which is basically at this point a byproduct, and the energy from the light is eventually, through some electron transport chains, going to end up on NADPH and ATP. So these are high energy molecules, but they're very short-lived, and we're going to need to use that energy and store it up, which is what plants are trying to do. So these molecules will go to the Calvin cycle, and this is found in the stroma. And basically what NADPH and ATP are going to do is capture carbon dioxide from the atmosphere. So they're going to take it from a gas form and eventually transform it into different types of carbohydrates. The last stage of this, of course, is the NADPH and the ATP are going to get recycled as their components. So ADP, inorganic phosphate, and NADP+. Here's an image of the light reactions from your textbook for just a little bit of a closer look. You can see that the light reactions are happening in the thylakoid membranes. So one thing we have here are two photosystems, photosystem 1 and photosystem 2. So it turns out that photosystem 1 was discovered first, even though it's the second photosystem, which is why it might be a little bit confusing here. But let's take a look at photosystem 2 because this is where it begins. So first light comes into the photosystems. And at photosystem 2, water is split into oxygen and also some hydrogen ions are released. So these hydrogen ions are pumped across membranes. We make a proton motive force, and when we have a proton motive force, often we're making ATP. So there is an ATP synthase to do so. So the ATP synthase uses the proton motive force, and it converts ADP and inorganic phosphate into ATP, and that is all sent to the Calvin cycle. So go back up to the where the light is going in, and you can see that the electrons are flowing from photosystem 2 to photosystem 1, and eventually those electrons are going to end up on NADPH, which also goes to the Calvin cycle and then eventually makes carbohydrates. Here's a summary of the light reactions from your lab manual to show you how today's assay will work. So there's lots of ways to measure photosynthesis. We could measure basically the end products of photosynthesis. So we could measure oxygen, we could measure NADPH production, we could even measure, in theory, carbohydrate production. What we are going to do today is measure photosynthesis indirectly. So we are going to measure the photosynthetic rate by mixing the chloroplast suspension with a chemical called 2,6-dichlorophenyl indophenol. We're just going to call that DCPIP for short. So in its oxidized form, DCPIP is a dark blue, but in its reduced form, it is colorless. So let's take a look at how that assay works. When mixed with the chloroplast solution, DCPIP accepts electrons as they flow down the electron transport chain of photosystem 2. Therefore, as photosynthesis occurs, DCPIP steals those electrons from photosystem 2 and results in the reduction of DCPIP and a color change from blue to colorless. We are going to measure this change in color from blue to colorless in our spectrophotometer by taking absorbance at 620 nanometers. Here is a photograph of the experimental setup. So basically what we have here is a lamp. The lamp is going to provide light. We also have in front of the lamp, we have a large beaker of water. And the whole idea there is that this lamp actually throws a lot of heat. So what I want to do is make sure that heat doesn't accelerate the reaction in the tubes, that the reaction is purely from light. And then over here on the right, I have some different beakers with coverings on them. So depending on what color we want. So for example, I have the dark control, it's covered in aluminum foil, and blue, red, and green all colored in different plastic filters so that we can get different colors of light. So here's our experimental setup. We're going to have 10 test tubes. The very first one is going to be a control. It's going to be there's going to be no light on it, so we want to see that photosynthesis does not occur when we put our test tube in the dark. So this is going to have 5 milliliters of chloroplast suspension and 150 microliters of our reagent DCPIP. Then we're also going to do some blue test tubes, so using blue light, green light, and red light. Each of these, notice we have 3 test tubes for each of them, the blue, the green, and the red, and this is because we are going to add some additional controls. So the blue test tubes are going to have one with a full reaction, so chloroplasts and the DCPIP, just like the dark reaction. We're also going to have a control where we're not going to have DCPIP. We just want to make sure that the chloroplasts themselves don't change color as they age over a 3-minute period. We're also going to have a control with no chloroplasts, just in case DCPIP changes naturally. Again, a good control where we're just removing one of the variables and testing to make sure that it doesn't react unless it's under the experimental conditions. Then we're going to do the same for green. Then we're going to do the same for red. So really we have 10 test tubes here, a lot of controls, and that's part of the experiment is subtracting the controls later on. So here are all the test tubes. I've set them all up and measured the 5 mils of chloroplast suspension or 5 mils of sorbitol buffer. So there's the dark control there. I have 3 test tubes for blue. The blue has 1 test tube with no chloroplast, so sorbitol only. It has 1 test tube that we are not going to add DCPIP to. We're going to do the same for green and the same for red. There's our Spectronic 200, and before we get started with the experiment, I need to calibrate it. So if you remember what that involves, it means I have to fill a cuvette with some buffer. So there's some sorbitol buffer. It's going to go into the instrument, and I'm going to press the 0 button. So you can see right now it is calibrating. The other thing that I did was I set it at 620 nanometers, and that is where it should be at, and now we're ready to start with the experiment. Let's get to our experiment. Now normally the experiment would be done entirely in the dark because stray light from the room will affect the results of this experiment. But what I'm going to do is demonstrate this for you with the lights on so that you can see how the entire thing works. So if you see here I have my test tubes, I have the DCPIP. What I'm going to do is I'm going to pipette 150 microliters of this. So if you take a look, there it is. It's a nice blue color. And I'm going to add it to my first test tube. So this one here will be the dark control. Next thing I'm going to do is vortex this gently. Quickly add it to my cuvette. Cuvette needs to be about 1/2, maybe 2/3 full. And then I'm going to take a measurement in the spectrophotometer. So here we go. I'm going quickly into the spec, get the reading right away. And there's my reading. So the next thing I'm going to do is I'm going to take it over to my apparatus. So of course, because this time I'm using the dark control, I'm going to select the beaker with the foil over it. So put that over top of my cuvette. Then I'm going to turn the light on, and I'm going to leave that on for 3 minutes. At the end of 3 minutes, I'm going to turn the light off. I'm going to grab the cuvette, and I'm going to take the measurement in the spectrophotometer. Now that I've done my dark control, I can work on test tubes 2, 3, and 4. So these are my blue experiments. So if you take a look, we have test tubes 2 and 3 have chloroplasts in them, and test tube 4 does not. So test tube number 2, just like what I did with the dark control, I'm going to add DCPIP to it, and then I'm going to vortex it and add it to a cuvette. So there's my cuvette, and just like before, I'm going to put one of the filtered beakers on it. This time I'm going to put the blue filtered beaker on it because this is one of the blue tests. I'm going to turn the light on, and I'm going to leave that exposed to blue light for 3 minutes, and then take my final measurement in the spectrophotometer. So once the first blue test is done, I'm going to move on to test tube 3. So test tube 3 has chloroplasts in it, but we're not going to add DCPIP to this one. This one is going to be water only, and it will be treated the same way. It will be vortexed, added to the cuvette, measured at time 0 and 3 minutes. Test tube 4 is a control that has no chloroplasts in it, but will have DCPIP. Once I'm done doing the blue test, I'm going to move on to test tubes 5, 6, and 7, and 5, 6, and 7 are going to use the green filter instead of the blue filter. Test tubes 8, 9, and 10 are going to use the red filter instead of the blue or green filters. So now I'm going to do this entire experiment for you in the dark. So I will collect all the data for you, and your job will be to analyze the data and make some graphs and form some conclusions for me. here's our data table, and I've done the experiment for you now. There is all of our data. So your job will be to do the rest. So if you take a look at this, I have times at time 0, I have readings, and I have readings at 3 minutes. So for each of those, you'll have to go through and do some math. So for example, in the dark control, I have 0.79 for 0 minutes and 0.78 for 3 minutes. So you'll have to go through and subtract 0.79 minus 0.78 and fill that in the column that says change in absorbance. You're going to have to do that for all of the colors and all of the experiments. If you're unsure how this works, please read the lab manual. There are clear instructions in there, and if you still don't know the answer after that, you can certainly check with me. So the last part you're going to do now is you have all that data, and you need to subtract the base data, and you need to subtract the controls away from it. So each of these you're going to subtract the dark control, you're going to subtract the chloroplast only control, and you're going to subtract the DCP-IP control. I am not going to go through an example with this because what I want you to do is to figure this out by reading the instructions in the lab manual. Once again, if you've gone over that and you're not 100% sure that you've done things right, you can check your answers with me, and you're going to end up with 3 numbers in the end. So one for blue, one for green, and one for red, and this will basically indicate the photosynthetic activity for the 3 different colors. Let's talk about the report for this particular lab. So there is the report grading sheet, and this particular report is a formal report. So notice my note here, it says this formal report is optional, and I will just say sort of. I'll get to that sort of in a moment. So why is it optional? It means you do not have to do it if you do not want to. You've already done one formal report, and you can do the second one only if you wish to do so. So if you decide to do Lab 7, that's great, and I will use the best grade. So the best grade between Lab 3 and Lab 7 to go towards your final mark in Biology 107. So if you did well, so if you did, let's say, greater than about 70 or 75, I would maybe encourage you not to do this formal report because you're probably not going to bring your grade up that much. If you did really poorly, this might be a good idea. This is an opportunity to improve your Biology 107 grade. As always, if you have done the report and you want me to look at a rough draft or something before the due date, I will be happy to do so. The more time you can give me, the better. If you just want to throw sections at me, like the introduction or something like that, I'd be happy to do so as well. So why did I say sort of? I said sort of because you probably want to at least go through the exercise and crunch the data, maybe make some rough drafts of the graphs because you're going to need to know how to do so for the lab exam. Yes, that's right. We have a lab exam at the end of the semester. So you do want to make sure that you know how to manipulate all the data and interpret all the graphs for that lab exam. We'll be talking about that in a couple of weeks. So speaking of graphs, this report will have 2 graphs. So part A, you're going to have a line graph that's going to be an absorbance spectrum. So on that graph, you are going to have the absorbance on the y-axis and the wavelength on the x-axis. For part B, you're going to have an action spectrum. So that is going to have the activity. This is from table 7-4. So the activity will be on the y-axis, and the color, so blue, green, or red, is going to be on the x-axis. So you're going to have those 2 graphs, you're going to have a wonderful report, and that is Lab 7.